Great. So is everyone from the room back here? All right, awesome. It does look like all of our contestants have now joined us. So welcome everyone. Welcome to the PFAS-Com. This is the second part of a community-based communication training and competition for trainees. So my name is Dr. Teresa Gillette. I'm an ORISE postdoc with the EPA, and I'm going to introduce our planning committee. So there is Anna Soul. She's a science and policy consultant from the Green Science Policy Institute. Shana Casper, water program director for the Community Action Works. Not able to join with us today, but an instrumental part of this program was Maya Fitzstevens, a research assistant with the Silent Spring Institute. Then there's Grace Campbell, a community engagement coordinator with NC State, Rebecca, a project director and ASSS Center for Scientific Evidence and Public Issues. And of course, Dr. Jamie DeWitt, professor at ECU. So thank you to our sponsors and organizers, the NCSU Center for Environmental and Health Effects of PFAS, the URI Sources, Transport, Exposure, and Effects of PFAS, the Green Science Policy Institute, Community Action Works, Silent Spring Institute, and the AAAS Epicenter. So really this project got started out of a PFAS conference hosted by Green Science Policy. There was a lot of discussion, and one of the things that I think I found to be really important, and I know a lot of other people do, is really how we communicate these complex issues to a community. As scientists and trainees, we're trained to really communicate during scientific conferences and to other educators, but sometimes we don't really have the best language or presence when it comes to a community, especially one impacted by the things that we are studying. And so with that, this idea was really started, and what we had envisioned was really training scientists and trainees to be able to communicate to the community. And so thank you all for joining us in part one. That was a really, really awesome, like, I guess, little both training from some of the people involved in PFAS research, as well as trying to give us some great examples of how scientists have really worked with communities to create more powerful studies and really what they're saying. And so with that, I'm going to ask our judges to introduce themselves. What we've done here, which is really different from a scientific conference, is we've asked community members and community advocates to actually help us judge these presentations that you're about to give. And so I think that's really important, because how we communicate to the public is really one of those things that should be highlighted more in science. And so I'm just going to call on the judges, and if you wouldn't mind just giving us a really 1 to 2 minute introduction about yourself and your particular community, that'd be really helpful. So I'm going to start with, how about Emily Donovan?
Hi, everyone. I live in Brunswick County, North Carolina. Chemours and DuPont dumped large quantities of PFAS into the Cape Fear River, which is our primary source of drinking water. We have probably some of the highest levels, at least that are being recorded right now, of PFAS in our tap water, per a couple of national studies that were done of some large water systems. We get our drinking water, like I said, surface water from the river. It feeds into 3 different counties. It impacts 300,000 residents. And this is actually, last week was the 4-year anniversary of us forming as a grassroots group. So we're all volunteers, and we just work to try and build accountability, as well as make our community whole and get lasting change. And I'm also on the leadership team of the National PFAS Contamination Coalition. Thanks.
Thank you so much for that, Emily. So Laureen Allen, would you mind going next?
Hi, I'm Laureen Allen. I live in Merrimack, New Hampshire. In Merrimack, New Hampshire, we learned in March of 2016 that Saint-Gobain Performance Plastics had been discharging via air emissions, which led to particulate that went into the soil, that went toward aquifers, also dumping into a nearby brook in the Merrimack River, also flushing PFAS chemicals into floor drains and entering many toxins into our sewer system, which gets put into compost and goes out to communities and et cetera, et cetera, et cetera. So we formed a group because after about a year of waiting for what we expected to happen, we had no idea that these chemicals were unregulated. The first thought of any community is, how can this happen? And how could this have gone on for so long? So, we've had quite a go of it, as many communities have, and we really appreciate the science and academic partners and allies that we have met. As Emily said, I'm also part of the PFAS Coalition leadership team, and we've done a lot of good work together, and we will continue to do so. So welcome to this group of scientists who's going to present to us. Yay, thank you.
Thank you, Laureen. Would Andrea Amico go next, please?
Sure. Hi, everybody. My name's Andrea Amico. I live in Portsmouth, New Hampshire, and my family's been personally impacted by PFAS contamination from the former Pease Air Force Base. The source of PFAS was from firefighting foam used not only at this military base, but at hundreds of military bases across the country. And I learned about our contamination back in 2014, co-founded a community action group called Testing for Peas, and also am part of the National PFAS Contamination Coalition. And I'm actually really looking forward to this event tonight and hearing what you have to say and being able to share constructive feedback so you can be armed with helping communities like mine in the future. Thank you for having me.
Thank you. And with Lemishia, sorry if I'm saying this wrong, Lemishia Kaminsky go next?
Hey there, good afternoon. So Lemishia Whittington Kaminsky, or as the community calls me, LA. Really good to be here, and thanks for having me. Really excited to hear the new current generation. Some of you are already doing the work, but of scientists that are really going to revolutionize the impact that we need in our community. So just really excited to be here. My work with PFAS began in the wake of Hurricane Florence. I'm an Afro-Indigenous woman from North Carolina, have worked in community for many years. I've had family that actually retired from DuPont, now Chemours. And so the impact of being within my family, but also working community, Hurricane Florence really exacerbated the spread of PFAS in a lot of our homes, our waterways, in addition to groundwater. And so that work entailed community education, workshops, seminars, and now leading a lot of national efforts alongside some of my co-conspirators on this call. So just really good to be here. Serve as Deputy Director of Advanced Carolina, Campaigns Director of North Carolina Black Alliance, and a Community Liaison with the National Academies of Sciences on their recent PFAS study alongside Emily. But good to be here.
Thank you. Would Dana Sargent from Cape Fear River Watch please go next?
Hi, everybody. I'm Dana Sargent. I'm the Executive Director at Cape Fear River Watch. Emily's already kind of summarized our PFAS issue. When we found out in 2017, I was working in communications, and since then, for the volunteer organization that Emily is leading currently, and then I transitioned into working with Cape Fear River Watch at that time. Excuse me. My background is in communications and then environmental policy. So I'm extremely excited. The issue of communicating science is just a piece that has always been near and dear to my heart. And I'm really excited that the leadership here, your professors and all of these folks here are putting such an emphasis on this. And I'm really excited to see what y'all have to share. My personal story relative to PFAS is that once we found out in June 2017 about our contamination crisis, and I was working, to learn about it with everybody else, I also found out a few months later that my brother, who is a firefighter from Chicago, was diagnosed with glioblastoma. He passed away in 2019 from that, and so I pay a lot of attention to the firefighter exposure. He was also a U.S. Marine. So that is sort of my personal impact. I also serve on the community liaison team that Lamisha had mentioned, and along with all the other things that we're doing, all of this just really relies on the scientific work. So I appreciate what you do.
Thank you so much, Dana. And finally, would Janelle Kimbrough please give us a quick introduction?
Good evening, everyone. My name is Janelle Kimbrough. I'm the Executive Director of Sustainable Sandhills. We are the mid-basin point of contact for PFAS work. Chemours, which is the company that released Genex into the Cape Fear River, is right in the middle of our reach area around the Cumberland-Bladen County border. And we have hundreds of private wells that are impacted by the PFAS release in a radius around the plant. So we help NC State, both the Community Education Corps and the Human Health Study with recruiting volunteers and getting information out to those affected homeowners and business owners in the area, making sure that they have the right information that they need. And we are really happy to be a part of this. So thank you for having us.
Thank you. Well, some brief logistics of tonight's event. Please note that this session is being recorded and remain on mute during the presentation talks. Please also enter any questions or ideas that you have in the chat, or raise your hand to indicate that you would like to ask your question verbally. So with that, I think it is about time to actually get started at this competition. Our first contestant is going to be Kaylee Kirkwood of NC State, and I'm going to ask Grace to yeah, there we go. So she's going to be doing PFAS profiling using pine needles. And so I am going to hand it over to Kaylee to get started. So thank you.
Thanks, Teresa. So what were the first questions that popped into your head when you learned about pollution in your community? Maybe something along the lines of, is it in my drinking water, my food, and the air I breathe? Or how will it impact my health and the health of my loved ones? I'm Kaylee, like Teresa said, a chemistry student at NC State University. And I had the same questions when I first read about PFAS contaminating the drinking water where I grew up in the city of Wilmington, North Carolina. At that time, and still to this day, most of the PFAS research has really focused on the health impacts of PFAS exposure and the levels of PFAS in food and water, which of course is very important, but there hasn't been nearly as much research on PFAS in the air. That's because airborne PFAS are a lot more difficult to study. It's not like food or water where you can easily collect and test hundreds of samples. And the equipment that you need to collect air is very expensive and in limited supply. But we know that PFAS can travel through the air. So my lab really wanted to understand more about that. And instead of building and using this expensive sampling equipment, we actually used pine needles as sort of natural samplers. Pine needles can capture PFAS and other chemicals in the air because of that waxy, sticky coating. So we went out and collected needles from all over the state. And you can see on this map that we get essentially a fingerprint for each location that shows the PFAS in the air at that site. And you can think of these circles kind of like pie charts, where each slice is going to be a different PFAS chemical, and the height of that slice is going to show the amount of PFAS at that location. So you can see that there are some areas that really stand out with very high levels of contamination, like the fluorochemical manufacturer Chemours, 2 airports, and a military site. And then the colors represent the different types of PFAS, and those are used for different reasons and have different sources. So you can see that the fingerprints are pretty different colors for somewhere like Chemours that produces a lot of these newer emerging PFAS chemicals that are colored in yellow and green. An example there would be Genex, compared to somewhere like the RDU airport and the National Guard that uses a lot of those older legacy PFAS chemicals that are colored in pink and blue here for uses like airplane coatings and firefighting foams. So this is really just the beginning of this project. Lucky for us, pine trees are extremely common across the state and really across the country. And so our goal is to expand this project to more locations and to actually get citizen scientists involved in collecting the needles so that we can help answer some of their questions. Thank you.
Wonderful. Thank you, Katie. So I think now we're going to break out to our networking room for the contestants, and the judges will stay in the main room. Hi, so our next contestant today is going to be Thomas Jackson, a graduate student from North Carolina State University. His talk is called Determining the Affinity of Proteins for PFAS. So take it away, Thomas. And Grace, please go to the next slide.
Thank you, Teresa, and thank you everyone for this opportunity to share some of my science and improve my community engagement skills. I am excited to talk about a method we have developed to measure how proteins bind PFAS. And the data that we gain using this method can provide information that helps answer the question, which PFAS are toxic? I'm a toxicologist, so my goal is to understand if PFAS are toxic to humans or animals. If they are toxic, I want to understand what organ systems are involved and how these chemicals can be toxic. For PFAS or any other potentially toxic chemical to cause harm, they have to enter the body and then be distributed to specific organ systems where they can accumulate. There are proteins that are responsible for this transport throughout the body, with the primary transport protein called serum albumin. In this top image, you can see a protein in blue, like albumin, binding PFAS and then being transported through the blood supply to organs like the liver. Albumin can bind lots of compounds, including essential compounds like fatty acids. And when we look at the structure of a fatty acid, like decanoic acid shown here in the black box, I want you to notice that it's almost identical to the PFAS below it, except for those hydrogens in the grayish color are replaced by fluorines in pink. Considering these similarities in structure, it's likely that PFAS are bound similarly to fatty acids by albumin, and that tight binding of proteins to PFAS can be a factor that lets them accumulate in organs where they can exert their toxic effects. When faced with most new compounds, toxicologists would measure toxicity in cell culture systems or look at epidemiological studies that associate the levels of a toxicant in blood with health outcomes, or carry out experimental studies in model animals like rodents. But unfortunately with PFAS, there are thousands of unique chemicals, and each have their own properties. They vary in size from 3 or 4 carbons to 13 or 14 carbons. They have different functional groups at the end. So they might be different acids, different molecules, different elements. And every one of those different combinations behaves differently in a biological system. Assessing every compound for potential toxicity would require many careers over many lifetimes and be extremely expensive. Meanwhile, people and animals will continue to be exposed without knowing what potential harms exist. We have developed a new rapid method that can screen PFAS, lots of PFAS, for binding with proteins using tools available in most labs. The results from this method can help us to prioritize the most concerning PFAS for further evaluation and can help us to determine what components of these chemicals might be causing the most harm. Based on the data we've already collected, we see albumin binding many of the PFAS found in the human exposure studies like the Gen X cohort in Wilmington, showing that this method can be a critical first step in evaluating the potential health effects of these diverse compounds. Thanks, everyone.
Thank you, Thomas. That was great. Okay, well, the contestants are going to break out again. And thank you, judges. If I'd actually add myself. So our next contestant today is going to be Drake Phelps, a graduate student from North Carolina State University. He is using zebrafish for rapid identification of the toxic effects of PFAS. So Drake, take it away. Thank you.
Thanks, Teresa. Appreciate that. Hi, everyone. Thanks for being here. My name is Drake Phelps. I'm excited to share some of my research with you here tonight. We've heard a few times tonight about how 4 years ago, the people of Wilmington, North Carolina, woke up to the news that their drinking water was contaminated with PFAS from a fluorochemical manufacturer. Researchers recognized this crisis for what it is and immediately went to work to determine whether these chemicals were in the blood of the more than 250,000 people that rely on this water every day. Last year, the GenX exposure study found that this community had levels of PFOS and PFHXS that were 2.5 times higher than the national median. Naphthalene byproduct 2 was also detected in the blood of this community, and before this compound had never been detected in human blood anywhere else on Earth. This community is still searching for the answers of what this might mean for their health. In my research, I used zebrafish to ask questions about how PFAS cause their toxic effects. One of the reasons I use zebrafish is because they develop so quickly. In fact, if we look at the top right box, we can see that they go from a single cell to a freely swimming fish in just 4 days. The data I can get from a zebrafish in a week would take about a month to obtain from a mouse or a rat. This makes them a great model for rapid identification of toxic chemicals. Zebrafish are freshwater fish, so not only can we use them as a model for fish that live in the Cape Fear River, but their genetic similarity to humans also makes them a great model for our own health as well. If we look at the bottom panel, we can see what a normal 4-day-old zebrafish looks like on the left. However, when exposed to the 3 PFAS I just mentioned, I see malformations in these fish, which I've labeled with the colored arrows. They have curved spines, smaller eyes, and swelling around their yolk sacs, which is where all of their nutrients come from during this part of their life. I also observed that these animals are smaller than the unexposed animals. This could potentially be a link to similar observations that other researchers have made where PFAS have been linked to low birth weight in humans. Overall, what I'd like you to take home is that zebrafish are an effective way to quickly identify the toxic effects of PFAS on developing animals, and we can use them as a model to protect the health of wildlife and humans. Thanks, everybody.
Thank you, Drake. Okay, great. Well, we're going to go back to our networking room, and thank you again, judges. Everybody. So next is Samantha Hall. She is a graduate student at Duke University, and she's going to be talking today about measuring and understanding PFAS chemicals in an exposed central North Carolina population's drinking water and blood. So Grace, if you can put the next slide up, and Samantha, it's all up to you.
Hi. Our lab has been analyzing water from the Hall River for PFAS contamination. And what we've seen is that PFAS levels in this river water can be relatively high and also really variable over different seasons. This river contamination is a concern because the community of Pittsburgh, North Carolina currently draws its drinking water from the Hall River, and treating the water doesn't remove these PFAS. So our group embarked on an exposure study to see if we could better understand how these PFAS levels in drinking water would relate to the PFAS levels in the blood of Pittsburgh residents. So we had about 50 people living in Pittsburgh graciously give us samples of both their drinking water and their blood at two different time points. And we analyzed all of these samples for PFAS. On the left, you can see the results for the drinking water, and there's quite a range in the distribution. So while some water samples had no PFAS, other water samples were over 400 nanograms per liter for total PFAS. What I really want to highlight is that the most frequent PFAS in this water were those in the purple and the blue colors, or the shorter chain C5, C6, and C7 acids. Because when we go to the blood samples on the right, we don't really see those compounds. We see a little of the C6 acid, but the top 3 PFAS that we saw in the blood were PFAS, PFOA, and PFHXS. And if we go further and compare these Pittsburgh blood levels to the general population, represented by that bottom dashed line, we see that our Pittsburgh participants have PFAS levels in their blood at about 2 to 4 times higher than the general population in the US. You can also see that Pittsburgh PFAS levels are similar to what's seen in people further downstream in this same watershed in Wilmington, North Carolina, which is represented by that top dashed line on those blood graphs. And so what we think explains these data is that based on previous studies of the Hall River, PFAS and PFOA levels in the water used to be much higher. So even though the levels of those specific PFAS are lower in the water now, the blood levels are still elevated from water the years before. And although the drinking water is higher now in these shorter chain PFAS, we're not currently seeing those build up in the blood quite like PFAS and PFOA. So big thanks to all of our study participants, and thanks to you for listening.
Thank you, Samantha. That was very interesting. Now we're going to break out to our next networking room. So thank you. Thank you. No, I love this title. Okay, so our next contestant is Anna Boatman. She's a graduate student from North Carolina State University. She's talking today about Bottoms Up, a laboratory system for studying PFAS accumulation in aquatic food chains.
All right, so probably all of you have had the experience in the summertime of walking next to a lake or a stream, and suddenly you find yourself in a swarm of tiny insects. Lucky for you, they were probably midges or maybe mayflies, and they don't actually have mouths as adults, so they can't bite you. Unlucky for those insects, they are probably about to be eaten by something else. So today I'm really excited to share with you how my lab is studying human exposure to PFAS by looking at aquatic insects. So aquatic insects, such as those midges and mayflies, also mosquitoes, dragonflies, they just mean any insect that spends part of its life cycle in the water. And they play a really important role in freshwater ecosystems as a critical link in the bottom of the food chain. So insects eat algae sitting at the very, very bottom of the food chain, and then insects get eaten by fish and other organisms higher up the food chain. So we know that PFAS have been found in freshwater streams in North Carolina and all around the world where these animals are living. We also know that PFAS has been detected in fish and insect and other animal tissue as well. And additionally, we know that certain types of PFAS are detected at higher levels in animals higher up on the food chain, which suggests that PFAS levels might be accumulating the higher up the food chain you go. But we don't have that data for very many PFAS, unfortunately. And that's where my lab comes in. So as you can see in this image on the right, we are able to simulate those bottom couple of levels of the food chain in our lab. So we can grow algae in water that we contaminate with whatever PFAS we want, and then feed that algae to mayflies that we also grow in the lab. And then we can measure how much PFAS get into the algae and mayfly tissue. And what we think we're starting to see is that some types of PFAS are going to accumulate more than others. Some PFAS might not be so bad. And we think that we'll also start to be able to see patterns based on the chemical structure of different PFAS. So as you know, PFAS are a very large class of thousands of chemicals. So if we can make predictions based on the chemical structure of these PFAS, we'll not have to test thousands of chemicals. We'll be able to see which ones, based on their chemical structure, might accumulate to higher levels. So what I hope you all can take away today is that the bottom of the food chain, those algae and insects, are super important, and studying them can help us understand which different PFAS might be riskier to accumulate to higher levels in people. Thank you.
Great, thank you so much. So we're gonna go back to our breakout room and we'll see you judges in a bit. Thank you. Thanks everybody for coming back. So next is Matthew Farrel. He's a graduate student from North Carolina State University, and he is using zebrafish to predict toxicity of novel PFAS.
Okay, hello everyone. So when you've been reading or listening to information about various PFAS so far, you've probably heard a lot of the same concerns come up over and over again, that they're everywhere, that they stick around for a really long time, that they're not very well regulated, and that there are thousands of them and more every day that we know very little about. And so my research is trying to more directly address that last point there, using zebrafish to test the toxic effects of these PFAS. So zebrafish are sort of small, common aquarium fish, about an inch or two long. But more importantly, for the research end of things, they produce a ton of eggs very quickly, and those eggs also mature into free swimming animals in just a couple of days, and past that only take a few months to reach adulthood. So the combination of producing lots of eggs and growing up quickly means that we can use zebrafish to study a ton of different PFAS chemicals over the full span of an organism's life, much more quickly than we would be able to using any other method, which lets us get a much better understanding of sort of what these chemicals can potentially be doing at different stages of life. Of course, there are so many of these chemicals that even with the high throughput testing we can do with zebrafish, it's still impossible to test all of these chemicals to a satisfactory degree. And so to that end, the goal of my research here is to not only test a number of PFAS chemicals, but also to test select groups of them so that we can figure out which specific characteristics of these different PFAS are causing toxicity. There are a number of different characteristics that differentiate one PFAS chemical from another. So some may be shorter, others are longer. Some are shaped like a single chain, whereas others split up and branch out a bunch. Some can be fairly strong acids, others not so much, and a ton of other characteristics as well. And so my hope is that through testing these chemicals on zebrafish, we can eventually narrow down which of these characteristics are linked to toxic effects that we see in the zebrafish. And by figuring out which specific characteristics of chemicals are causing toxic effects, we can then in the future, as chemical companies are developing more and more of these and dumping more and more of them into your drinking water, we can basically use this testing to narrow down exactly which ones are going to be dangerous and need to be more strictly regulated in the future, which will hopefully allow us to do some proactive regulation rather than just constantly playing catch up. Thank you.
Thank you. That was great. So we're going to go out to our breakout room and we'll see you guys later. Thank you. And thanks again for joining us. So Chrissy Crew, a graduate student from Duke University, is going to be giving us our next talk. And she is saying, is PFAS contaminated drinking water putting North Carolina pregnancies at risk? So here's Chrissy. Thank you.
Thank you. If you are told that you have a man-made chemical in your blood that isn't really supposed to be there and is in concentrations 4 times higher than the average population, I guess your first thought would be, well, what does this mean for my health? And additionally, if you're a woman who is pregnant, your next question would probably be, well, what about the health of my baby? If this were me, I'd be concerned and then pretty frustrated when I was told there's very little known about what these blood results mean for my health. So this is where my research began. I work with a team of scientists, you heard from my colleague Samantha Hall, who has recently identified uncommonly high levels of PFAS chemicals in the blood of community members in Pittsboro, North Carolina, which is close to my home. I'm one of the scientists who's working to understand what this means for community health. Specifically, I'm studying how a woman's exposure to these chemicals affects the health of her and her baby during pregnancy. Why do I look at pregnancy? Well, as any mothers here know, pregnancy takes a huge toll on the mother's body and can put her at higher risk for diseases. It's also a critical time for healthy development of her baby. So I'm trying to understand if PFAS exposure during pregnancy puts the mom and baby at extra risk for poor health outcomes. So to do this, I'm using an animal model so that I can determine health outcomes resulting directly from PFAS exposure when all the other factors are controlled for. What's important about this experiment is that many previous studies just look at one type of PFAS chemical, where this experiment looked at a mixture, which better resembles human exposure. Additionally, the rabbit drinking water has the same PFAS exposure as a Pittsboro household. So this experimental exposure resembles what a human is taking in. And out of many animal models, the rabbit pregnancy very closely resembles human pregnancy. So I gave the rabbits PFAS contaminated drinking water before and throughout their pregnancy. And then I evaluated health outcomes located in the blue box on the slide to determine how the PFAS exposure impacted mom and baby's health. One interesting result we found so far is that PFAS exposed mother rabbits have increased blood pressure. And some of the blood chemistry tests showed possible liver and kidney dysfunction. Together, these results are suggestive of preeclampsia, a pregnancy complication that is the leading cause of maternal and fetal death in the United States. So this is an important finding because early intervention of preeclampsia is critical for the best health outcomes. So my research suggests that mothers in Pittsburgh should be made aware of the warning signs of preeclampsia and work with their doctors to monitor symptoms throughout pregnancy. Educating women in this region about the potential risks of their pregnancy is a critical next step to protect the health of mothers and their babies. Thank you.
Thank you. So we're going to go out to our breakout room and we'll be back in a little bit. Thank you, judges. Great. So our next contestant is Matt Dunn. He is a graduate student from the University of Rhode Island, and he's going to be talking today about a simple solution to a complex problem.
Awesome. Can you go to the next slide? Thank you. So you might notice on my slide that there's a lack of chemicals, there's a lack of data and a lack of figures and fancy shapes that none of us really like looking at. Well, that's actually by design, because the whole purpose of my research and my PhD is to develop a tool that makes us not really have to worry about those things too much when we're trying to detect them, monitor them, and measure them in our surface waters. So it's supposed to be just as easy for me, a scientist, as it is for a community member. And at the end of the day, the goal of my research is to actually create something called a passive sampler. Now that sounds like jargon, but it's actually exactly what it sounds like. It's passively sampling the water. No power source, no battery, no mechanics, no maintenance. So let me try to describe it to you like this. If you told me you wanted me to come to the Cape Fear estuary and measure PFAS in the surface water, would you rather that I come and take 1 liter of water on one random day and take it back to the lab? Or would you rather that I come every single day and take 2 milliliters until I have a full liter over the course of a month? What would give you a better picture of what's actually in the water over the course of a month feeding your drinking water? Obviously, it would be the 2 milliliters each day averaging out to what you're actually exposed to. That's exactly what this little tube here does. Using a lot of math and chemistry and science that even I don't enjoy, we've been able to design and sort of validate, so to speak, or work on a way to prove that it works, that this can do exactly that. Take a little bit of PFAS from your water in your groundwater well, in your surface water river that's feeding your drinking water, in an estuary, an ocean, a pond, a lake, whatever surface body of water you're concerned about. And the real convenience of this is that through proving this mathematically and doing all that chemistry, what I really want to focus on is the convenience, because I am hoping to have at the end of this research, my PhD, something I can mail to you and say, you don't need to be a scientist. Take this out of the tube, strap it at the end of your dock, strap it at the end of the river, at the end of your street, go back a month later, mail it to me. And now I can sample your water, give you much better, broader view of the contamination of PFAS, and do it honestly much cheaper and with much less labor than traditional methods, because many of you might not know this, and many of you might. Trying to actually measure PFAS in a water sample is not only very expensive, but it takes a lot of time. It takes a very complex laboratory. And it's no fun also, it's very laborious. This is much simpler. It requires only one scientist mostly instead of a team of scientists. And it really reduces the things that we need that communities don't have access to, things like solvents, big complex labs, sample bottles, freezer space. It's just much more convenient. And we're using a lot of math and engineering to prove that in over month-long deployments. And that's really how I wanted to focus on sharing my research, is that here's a tool that I hope to be able to mail to you in a couple months and be able to give you a very broad picture of the PFAS contamination in your water. So thanks.
Great, thank you, Matt. So we're gonna head on to our breakout room and we'll let the judges judge. Hi, welcome back, everyone. Well, we have the results from the judges. And so in second place today is Anna Boatman. Thank you so much. And then first place is going to be Chrissy Crew. So woo, congratulations to our winners. And if you can see in the chat, there's going to be a specific one for voting for the Participants' Choice Award. So please go and do that right now so that we can look at those results. But I just want to give a really big shout out to all of our competitors today. Wow, just so impressive. I learned so much, and I have to say, like, I'm very, very, very proud of the younger generation and what you're going to be bringing to science and PFAS science in the future. So thank you so much. I also want to shout out to our judges. Thank you for taking the time to do this. We really, really, really appreciate you being able to give us some great feedback about basically how we're doing. So thank you. And now let's see if we have the results yet. Do we?
So Shane, are we ready for the Participants' Choice?
Yes, so it looks like we have a winner, and the winner for the Participants' Choice is Kaylee Kirkwood. Yay! Congratulations, Kaylee, and congratulations to all of our participants today.